Hello everyone, my name is Dohyup Kim. I am from the Van Haren lab at the Stanford University School of Medicine. And I'm here to talk about the clinical update on phase 1 trial of vitamin D in ALD boys. So beginning with the funding sources, this huge project has been only available from the initial trial funding from NIH and the National Institute of Neurological Disorders and Stroke. And also the huge part of this project involves in the single cell sequencing, which is a top-notch state-of-the-art technique of sequencing funded by the Broussard family and the Adler family, and also the trial funding. And my salary has been paid by these great families, St. Harrison family and the Leonard Yola family, and also the Stanford. Also, there are a lot of people who are involved in this project. We have an awesome lab and Heron lab at the Stanford Department of Neurology. And also we have a lot of collaborators across our school and also across the state and across the world. So I appreciate them. So to start, we all know about the ALD. It's a disease caused by the mutation called in gene called the ABCD1 gene. It's involved in the very long chain fatty acid metabolism. So actually this gene encodes for the protein transporter, which will let the VLCFA fatty acid chain into the peroxisome so that it can degrade. But when we have a mutation of this gene, the function of this protein has a problem so that the very long chain fatty acid accumulates in our body, and it will give us the reactive oxygen stress, which will result in mitochondrial dysfunction, and also have a lot of stress on our ERs. It will lead to the cell death. And also this accumulation of very long chain fatty acid will lead to the inflammation in our body. So together, we have a neurodegeneration in our brain and also the adrenal insufficiency. There are several types of ALD. The most severe form is a cerebral ALD. It has a progressive neuroinflammation and neurodegeneration. And if we actually see the graph of when it happens, is that CALD, cerebral ALD, usually happens in childhood. 40% of the ALD patients show the phenotype of CALD at around the age of 5 and 6. And then the remainder of the patient CALD phenotype happens in their adulthood. And we have a pilot study of vitamin D treatment in boys with the X-linked ALD. Our goal is to establish the dosing regimen of the vitamin D so that we can know how much of a vitamin D that we can give them to have a preventive or delay the progression of this ALD. So our target of the patient was 20 patients. And then what we do is that we give them the dose of vitamin D over the course of 12 months. And then right before we measure different phenotypes like vitamin D level, MRI, MRS, and IGSH. And then over the course, we measure those phenotypes again in the middle at 6 months. And then at the end, after 1 year, we measure them again so that how they are changing and how effective vitamin D are in our treatment. And the enrollment period started back in 2016 and ended in 2019. We got our data in the middle of 2020, and we're actually at the beginning of processing and analyzing our data. And those data was collected from 2 different places at our facility, Stanford University, and also at the Back East and Kennedy Krieger Institute. But at the same time, what we did was that we measured the molecular level activity of those vitamin D treatment. So our goal is to not only to look at how much that we can give them to have a best result, but also we want to investigate what's happening in their body and what vitamin D actually does in their body to delay or prevent the CALD happening. So we also measured at the same time single-cell RNA sequencing, metabolomics, and lipidomics and meta-array to see the molecular signature of those patients. So my job is to get those data and analyze those and trying to investigate what's going on in their cell. So quickly, the patient demographic is we luckily have 21 participants included in our data, and their median age was 6.7, and they have various race and with the adrenal insufficiency and the vitamin D level. The initial data that we got is the vitamin D level in their blood. So baseline, they have a relatively lower vitamin D level in their blood. But as soon as we start the vitamin D treatment, we see the increase of vitamin D level in their blood. So the vitamin D treatment is actually happening, and we want to find out what it does. So the overall direction is that we are measuring metabolomics, single-cell RNA sequencing, and lipidomics and epigenomics as well. And this is in collaboration with 2 other labs. So metabolomics analysis and the single-cell RNA sequencing assay has been done by the Snyder lab, and also the lipidomics has been done by the Kemp lab. All right, so first, today, so I'll be talking about these first 2, because we just got these data, and we're in the process of preliminary analysis of the metabolomics and the single-cell RNA sequencing. So first, we are looking at the metabolomics analysis. So just quickly, for those who are not familiar with what metabolomics are, so here is what's happening in our cell. So we have the gene or the genome with the DNA, and then DNA it encodes for RNA, which will be translated into proteins, and then it will have the phenotypic effect, and it will have effects on sugars, nucleotides, amino acids, and lipids. And then it has a direct association of what's actually happening in our cell. So when we are looking at the metabolomics, we are actually looking at the forefront of molecular level of what's affecting our body. So the first thing that we got when we got the data is to compare those metabolite profiles against the healthy control. So this is the PLSDA plot. It's similar to PCA plot, but we can actually see the profiles of metabolites of the XALD patients in red have a distinct grouping compared to the age-matching healthy people in green. So they do have different metabolites in their cells. And when we are looking at the top 50 metabolites, we can actually see the differences better. So here on the left, on the top, the red group is ALD patients. On the right, green is healthy control. And we can see the list of these metabolites that are more abundant in healthy control in red, and less abundant in the ALD patients in blue. So we can actually measure and point out what metabolites are different between two groups, and then we can study better, understand better what's going on in our cell. So I also, since we have 3 different time point data, so I can actually plot them against each other as well. So we can see the different groupings of different visits. Visit 1 is 0 months, right before the vitamin D treatment in red. And then in the middle, we have 6 months, middle data point. And then we have another grouping, type grouping of visit 3 at the end of the vitamin D treatment. So it's actually shifting upwards, that the metabolites in their body is changing. If I look at the metabolite, top 50 metabolite, well, actually top 100 metabolites, again against the different visits, you can actually see the different metabolites are behaving differently between visit 1, 2, and 3. And you can see the visit 2 and 3 have similar metabolite profiles, which actually coincide with the vitamin D level that we saw earlier, that from the start, the early onset of the trial, the vitamin D level increases. So it is actually matching what we're expecting to see. And we are also interested in how these are different from the healthy people. So I also added the healthy control. So maybe it's just me that's seeing it, but from the visit 1 in red to visit 2 in green and visit 3 in blue, it is actually going towards the healthy control in cyan. The data is, since the data points are too large, it maybe just, it's a little bit noisy, but we are expecting to see, as we treat them with the vitamin D, that the metabolite profiles in their body is getting closer to the healthy people. All right, so the metabolites profiles are important, and our next step is to look at individual metabolites to see which one is affecting the most. And that's our next step. And we are now going next to the single cell RNA sequencing. So single cell RNA sequencing, actually the RNA sequencing, for those who are not really familiar with the RNA sequencing, is that RNA is in the middle between the DNA and the proteins. It's a middle component of turning into proteins. And what we can do with this RNA sequencing is that we have 2 different conditions, tumor conditions in this. In our case, it's an ALD condition and the normal condition. We can measure the RNAs in our samples and how much of these DNAs are expressed, how much individual genes are expressed in our sample. So we can compare these 2 samples together to see which genes are important in our condition and which genes are absent in our condition. So it's a great technology to look at the effect of the gene expression in our sample. And the single cell RNA sequencing goes one step further, is that it adds additional step of dissociation of different cells. So before, in the conventional RNA sequencing, we were only able to see the average level of all the samples that we put together. But it is actually, single cell RNA sequencing is actually putting apart each individual cells and measure which genes are how much expressed within our single cell. So what we can do is, while bulk RNA sequencing, the conventional RNA sequencing is only getting the average of different types of cells included in our sample, we can now actually see or divide into different types of cells and what genes are expressed in different types of cells. So it's a great technique. It is much more expensive. It is top-notch, but it gives a huge more amount of data and huge more amount of information that we can actually look at closer to what's actually happening in our body. So we finally got the result of single-cell RNA sequencing. We were able to sequence 310,000 cells. And after the quality control, I was able to analyze 170 cells of the ALD patients. And these different colors are visit 1, 0 months, visit 2, 6 months, and visit 3, 12 months. And they have similar but different clustering. So if we take them apart, they have similar clustering. So each dot are each cell. But each cluster have different distribution, which means that there are some different gene expression profiles or cell-specific gene expressions differences between visit 1, 2, and 3. So now what we can do is that what each clusters mean, what cell type it is. So that's what I was trying to do, is to identify the cell type of each cluster with different colors. And I was able to find the major immune cells here, the T cells on the top, a large cluster, and the B cells, natural killer cells, and monocytes, and the trace of platelets and red blood cells, and then some precursor cells. So now with the single-cell RNA sequencing, we can actually see what we weren't able to see before, what each cell types are doing. So the first thing that I did was that to look at the ratio of each cells in our sample. So T cells are over 40%, B cells and monocytes are over 20%, natural killer cells about 10%, and which is similar to the healthy people ratios as well. And when I actually look at the ratio of T cells across the visits, I was able to find that there's a decrease of T cell content and the increase of B cell content. So, so the huge difference was from the T cells. So I look at the T cells more closely that I was able to observe that the CD4 naive T cells are decreasing over time. So this might be from the treatment of vitamin D, or this might be from their age progression. So our median, remember our patient median age was 6.7, so that they're in their earlier age when their immune functions and immune cells change dramatically. So it may be from their age process, that's natural, or it may be from the vitamin treatment that we give them. So to find out which that is, we can actually, with the single-cell RNA sequencing, to look at the individual genes. So what I did was that I compared the genes from the right before the vitamin D treatment and right after the vitamin treatment at 12 months. So, and identify which genes are differentially expressed and how they are differentially expressed. So in each cell type, I was able to identify different genes, differentially expressed genes between the 0 months and the 12 months. So here, CD4 naive T cells have 55 upregulated genes at 12 months, meaning that after we give them vitamin D, those 55 genes are more expressed. They're more abundant compared to the beginning of the treatment. And there are also 96 downregulated genes as well. So this is a huge amount of data that some of the cell types have like over 600 genes that are differently upregulated, meaning that they have such huge differences between the vitamin D treatment. So it will take a lot of time for us to analyze all this data and to look at these genes and which are really important in our treatment and in their ALD disease. But give you the example of what we can do with this huge data is I picked out one gene from this this list. Is mTRNR2L8. This is a pseudogene of mitochondrial mTRNR2, means that it's actually the copy of mTRNR2 gene, which is located in mitochondria, and it encodes a protein called humanin, which has a neuroprotective and cytoprotective effect. It prevents the neuron degeneration and cell death. And this copy of the humanin, I was able to find that they are more expressed, much more expressed after we give them vitamin D. So the y-axis is the fold change from the 0 months to 12 months, meaning that in the CD4 naive T cells, you can actually see almost a threefold increase of this gene at the end of the trial. So most of the cell types have a fold change increase, where we can also see a few of them are decreasing. And also the ones that are missing is that they don't have the significant change. So, so this humanin protein, just really quickly, is a short polypeptide, meaning that this protein is really small, and it suppresses neuronal cell death caused by Alzheimer's disease. So it is heavily studied by the Alzheimer's Disease Society, and it is from the mitochondria, and it has a neuroprotective and cytoprotective effect. And we are actually looking at the copy of this gene. Is an isoform of mTRNR2, and it is overexpressed. And earlier study showed that you can find a lot of this gene in the brain tissue of Alzheimer's disease and the suicidal depression patients. And they thought, they're thinking that those proteins have compensatory mechanism to protect the neurons from the chronic stress. And we are actually missing some of the cell types. And another interesting thing that we could see was that this humanin have another copy in our genome, is mTRNR2L12. Is for those cell types that are missing the fold change of L8, and they are actually increasing. L12 is increasing, having an overexpression at the 12 months. So that was a little bit interesting. So L12 is another isoform of mTRNR2. There's not much of the study on these two, but it could be a target of what we can further study. But this gene, this example is just a snapshot of what we can see from the single cell RNA study. I'm not saying that this is the key gene of what's going on in the ALD patients and with the vitamin D, but we can actually see something like this for all other genes that I was able to identify, like those over 600 genes that are differently expressed. So we have this huge data and foundation of data that we can actually look at for us and for other scientists in the society as well. So the quickly conclusion is that we did the primary analysis of vitamin D trial. And we looked at the metabolomics, and it enables us to investigate the metabolomic profiles of ALD patients. And ALD patients have different profiles of the metabolites compared to the healthy control, and also different visits have different metabolite profiles as well. And also the 12-month metabolite profiles are more similar to those of healthy control as well. Also, the single-cell RNA, we were able to see the snapshot of RNA sequencing. So single-cell RNA sequencing enables us to have a broader and also the final look at the gene expression levels. And those gene expression profiles are distinct between different visits. And using the single-cell RNA sequencing, we can study what's going on in individual cell types and also individual genes, and what happens on each gene in different cell types as well. So I'd like to thank those members of our lab. We have a great lab, Manhattan Lab, who are doing wonderful research on ALD, and also this study wasn't able to happen without the help of our collaborators. So thank you so much for the opportunity to present our data, and we are about to have a great data result in our study, and more results will be coming as our data analysis progress as well.
Thank you so much, Dr. Kim. As a parent, vitamin D just seems so accessible, right? It's something that we understand and we know what it is already. It's not something we can't pronounce that we have to learn about. So it's always exciting to get updates on this topic. There is one question so far from Dr. Lund. Did any person with ALD develop cerebral disease on this preliminary trial?
So I don't know if you can see me in the spotlight here, but this is Keith in here, and Dohyup, awesome job presenting that. That was really great. To answer Troy's question, no, nobody who's in the trial has developed any lesions today.
Okay. And I appreciated too the map in the beginning showing the different centers for anyone who might be interested. Or is there any plans for expanding?
Yeah, thanks for queuing up the question, Miranda. So several of us in the ALD research space are putting together right now a very large follow-up trial, a phase 2 trial. I can just speak briefly to what it will look like. It's still in the proposal stage, going to be submitted to NIH. Our hope is that we'll open next year. It is a trial where people will be able to enroll at their home institution or their home doctor's office. They don't need to travel. So we're going to make it very accessible. And they will, to participate, you can be between 0 and 10 years of age with ALD, no brain lesions. And you send us blood work in your MRI reports, and we send you the vitamin D. And we give you a small stipend for participating every time you send blood work and every time you send us your MRI. And that will go on for a few years. We need a lot of participants to make it work. So it really will, for this to work, it'll actually be a really big community effort. And assuming we get funded, we will eventually, but whenever we get funded, you'll hear a lot more from me and probably from some of the other ALD nonprofits trying to encourage people and families to enroll. We're trying to make it very, very easy and simple, and so people can do it. You can do it from home. So it'll be very exciting in that way. You don't have to travel far away to some faraway place to participate in the study. So you'll hear more. The tentative name I'm using for the trial is OVID, meaning oral vitamin D in ALD boys. So OVID will be the name of the trial. And there'll be a lot to it. We're really excited about where this might go. We agree this is a potentially simple strategy that could be helpful. We don't know yet, but we hope it will be. And yeah, we'll be looking forward to having more people join in. And I think Eric may have a question.
Yes.
Eric?
Yeah, I'm very much looking forward to these future trials coming down the road and the work you're doing. I have a question though about the RNA-seq and the metabolomics. It's extraordinarily interesting, and I think it's where we need to go. Can either Dr. Kim or Keith comment on, as I understand it, you're quantifying what genes are upregulated and downregulated in certain immune cells, and immune cells function differently. You know, there's pro-inflammatory, some immune cell subsets are anti-inflammatory. So understanding, can you comment a little bit on when you see an upregulation in a gene that you think is neuroprotective, how are you interpreting that in the setting of what cell type it's living in? And how are you, or is there a way to control for other environmental factors that may account for that up or down gene regulation? Because immune cells are constantly coming into contact with our environment. When you get sick, your T cell populations change, your B cell populations change, albeit acutely. So it's a complicated question, but I think it'll help us narrow the signal as to what is maybe more specific to ALD than being a person living in an environment surrounded by microbes.
Yeah, that's a great question. First of all, I think your question is exactly why we're trying to do the single cell RNA sequencing rather than the conventional RNA sequencing. So if we see the conventional RNA sequencing, we can only see the average of different T cells. Even if we just take out the T cells from our sample, we can actually see the average of it. But if we see the single cell RNA sequencing, I don't know if you can remember from my slide before, but there are different types of T cells and different types of B cells that we can cluster, separate, and then look at what is going on in each type of T cells or each type of B cells. So it's a really powerful technique to see what really is going on in different immune cells. And that's why we are actually using the single cell RNA sequencing, that we understand there are different cell types within each type of immune cells as well. And so another thing is that the other factors that might be involved in immune cells that will affect the gene expression levels. That's why we need the participation of a large group of patients so that we can hopefully remove those variables that are not from the ALD disease, but we can actually see what's going on in the ALD disease as we have more sample data or more patient data, that we can actually see the true signal coming from the disease.
And thanks, Dohyup. And to follow up on the interpretation piece, so Dohyup was showing kind of an example of what we can do with this single cell sequencing, commenting on the significance of the genes, especially one that's obscure as the one we're talking about in the in this slide deck. I mean, there's nothing we can say about whether they're relevant to ALD, to vitamin D, or whether they might be related to vitamin D or not. This is really starting as purely observations that will then maybe be followed up in another model or in a future trial. Developing an understanding about whether one of these gene changes is due to vitamin D or whether it's even helpful or neutral or harmful is not something we'll be able to say from this study, but we will be able to try and get a sense of what did change, and then follow that up in mouse experiments or human trials or something similar in the future. And that might tell us a little bit more as we learn more about how ALD works and how some of the future therapies like vitamin D or other small molecules or gene therapies in the future.
Thank you. I just have one more question. So I know the families that are followed at some of the bigger ALD centers are more likely to find out about this, but how can we help you get the word out as you're looking to expand to as many families as possible about this, especially now that we can trial at home?
Yeah, well, we're hoping to have the trial set up. We need funding for it first. I will be reaching out to a few of the ALD nonprofits to looking for just letters of support for what we're doing with the trial. I didn't want to bother the ALD Alliance, for example, right before their session, but I have a letter that I'm going to send you after this.
How's Monday sound?
Yeah, sounds good. Great. But I think, yeah, when we do open the trial, we're going to be looking for more than 300 participants to try and join. So we'll need a lot of families. Yeah, I mean, I think one doesn't have to participate to take vitamin D, but it really does matter to the community and to understanding what's safe and what's not, how things work. If we understand if vitamin D works, which we all hope it will, understanding how it works matters a great deal to building new therapies. And also, if it works, it's going to be important to know that when we're running clinical trials for other treatments, and we'll need to, make sure we control for vitamin D, and so we don't have more people on vitamin D in one arm of the trial than another, or something like that. And these will be really important questions for us to answer now and in the future. And so I really do hope that when the time comes, and I hope in the next year or so, people will be willing to join our study, which we're making as easy as possible. But for now, stay tuned, and we'll have more soon.
One other question did pop up that I know the answer to, because I remember a great visual that someone presented last year. I think it was Dr. Bonkowski, but it might have been you, Dr. Van Haren. Has there been a greater incidence of ALD, cerebral versus non-cerebral, in places with less sunlight?
Yeah, Dr. Bonkowski's lab looked at this last year, and we have a publication that's hopefully forthcoming soon. But yeah, he found that at the higher latitudes, meaning higher up in the in the United States, where they get less sunlight in the winter, there were higher rates of cerebral ALD, and vice versa down south. So that would be circumstantial evidence that either vitamin D or sunlight is helpful.
Great. Thank you so much. Thank you, Dr. Kim, for your presentation. Lots of exciting developments.